Nobel Prize winner for what is it? This area is physiology, medicine, right? Went to 3 people who work on vesicular transport. One of them actually worked on the actual proteins, multiple proteins, but one of those proteins are the protein that pull the polypeptide into the ER. And that work I read about it from the late 1980s. So this is we're talking about relevant stuff here, and this is people get Nobel Prizes for this. Okay, so it's kind of interesting. Yeah, if you want to look up their names and papers and read about them. The other announcement is, I forgot, I should have said this at the beginning or at least after the first exam. I was just reminded, about looking at the key for each exam. Again, I don't mind you coming in. What happens in the past is that nobody comes in, as I was reminded that not too many people came in to look, and then until the last minute, until the last week, and then everybody pour in, looking at all kinds of key, and so it's just too chaotic. So what I'm going to do is, I will ask you if you want to look at exam 1 and 2. I will have them in Sequoia 120A. During office hours. There are 4 different office hours. You can go in there and look at them. I'll have them in there for another week or so, okay? And then after you take exam 3, I'll have exam 3 down there, and then you can look at exam 3 until the next exam, and so on and so forth, so that I don't want you to wait until the very last minute, and then that area will be just inundated with people, and you can't get more than 6 or 7 people in that room, and I don't want you to do that, okay? So please make yourself, if you're interested in looking at the keys, make yourself look at the keys early, okay? Often is okay too, but at least early, okay? Again, the keys will only be available for 2-3 weeks at a time, right? 2-3 weeks, and then that's it, okay? So sorry to do that to you, but I just have to regulate the number of people in the office. Okay, so we're talking about vesicular transport, which is the really last big idea. I should be able to finish and then have a study session if you want at the end. So very quickly, reminding you of this is vesicular transport involves budding, transport, targeting, and fusion of membranes. So the budding process requires cargo proteins, Clathrin type of cargo proteins, which then induce or carry out the actual budding part. That neck eventually will have the long protein diamond wrapping around it, and then sever the neck so that you have a coated vesicle. The coat there will block any lipid signature, any protein signature, we talked about lipid and protein signatures, right? Yes, okay. Lipid signature, protein signatures, as well as cytosolic submembrane protein meshwork. So the coat proteins need to be removed. The process of removing the coat protein is called uncoating, so that you have a naked transport vesicle. To be transported. Remind you that there are 3 different types of coat proteins. Each one is responsible for the budding from different membranes, right? And vesicular transport can go in both directions. Can go out this way through all the Golgi cisternae into the plasma membrane. It can also go in the other direction as well. Okay. So then the transport and targeting. Transport will be along the microtubule. And if the cell were transporting vesicles from the ER out toward the plasma membrane, then therefore it will be along the microtubules that, remember, they radiate out. So then that will be along the toward the plus end of the microtubules, therefore kinesin will be responsible. Once if let's say these vesicles somehow got moved, need to be moved toward the plasma membrane, remember the cell cortex also has not only microtubules but microfilaments. So these vesicles can actually move sideways as well. If they get moved sideways, then myosin can move them along microfilaments, but only within the cell cortex. Okay? So again, vesicles can be moved either along the microtubules or along the microfilaments, and then using different molecular motors. Remember protein signature, we talked about that.
I'm sorry, what did you say about myosin?
Myosin is the molecular motor to move materials along the microfilaments. So transport again. So what people call the head, which I kind of think about it as the feet, they would literally walk the vesicle either going that way or going this way. If the vesicles are moved toward the plasma membrane, it'll be by kinesin along the microtubule toward the plus end. If the vesicles need to move back toward the ER, then that'll be carried out by a dynein, right, along the microtubules toward the minus end. And we'll talk about lipid trafficking shortly here. Okay, just to show you, I think, didn't I show some of these pictures before? So just to remind you that, how do you know that dynein needs to be involved? How do you know that kinesin needs to be involved? How do you know that myosin needs to be involved? It depends really on the cytosolic submembrane meshwork, right? That's what they recognize. The cytosolic submembrane meshwork is so thick that the molecular motor will not be able to recognize any membrane proteins in here, will not be able to recognize the lipid signature. So it's up to the submembrane protein meshwork. That's why each membrane also contains its own specific cytosolic submembrane meshwork, protein meshwork. Okay, and the vesicle is the same. Okay, targeting. Targeting is when both Rab and snare, and I think this is roughly where we stopped here, is Rab and snare, snare, V snare, and T snare, as well as Rab. So V stands for vesicle, T stands for target. So there is a whole family of V snare, there's a whole family of T snare. Each one is present in a different membrane, right? And then V snare will be targeted, specific T snare, specific V snare will be targeted to specific T snare. And then once the vesicle is targeted to the organelle, that V snare and T snare will actually interact to hold the vesicle in place. Right? It has to be held in place for fusion to take place. If for some reason, let's say you have a mutation of the snare proteins, then targeting and transport can occur, but you won't have fusion because the vesicle will not stay in place long enough for fusion to occur. Okay? Yes, sir.
Okay. So when you have actual fusion, since like on the outer membrane you have the hydrophilic sides, right? Is there something that mediates the binding of the membranes?
No, we'll talk about
okay.
that. Fusion is very particular, specific.
Okay.
So we'll talk about that. Yes. And I think I know what your question is, and about the face and all of that. So we'll just kind of tease it out and look at it.
Okay.
Rab actually is, remember, all the snare and Rab indicates protein signature, right? So then there are different Rabs that then can be recognized by different, what we call Rab effector, basically Rab as well. So there are different Rabs all over the place, and only specific Rabs will be recognized and by specific Rab effector. Okay, so that's targeting. Okay. So now we're at this part where the vesicle now is held in place temporarily, this is not a permanent thing, temporarily, and then fusion takes place, and this comes to your answer. So how does fusion occur? Right, there you go. How does fusion occur? This is a very important slide right here. Fusion occurs between any two membranes that are a good match. That means that they are similar. They're not the same thing, they're just similar. And fusion actually occurs spontaneously. Because why? Because it's hydrophobic interaction. And hydrophobic interaction is fairly spontaneous. If you put two things that are hydrophobic close enough together, guess what? They're going to interact. So that answers your question. So therefore, the interaction between V star and T star is very important, because that then would allow the two membranes to not only be close enough together, but to be close enough together long enough for fusion to occur. And fusion is spontaneous, but only if the two membranes are similar. Back many slides. That explains why that vesicle from the ER can only move toward the cis face of the Golgi and nowhere else, is because the two membranes here are more similar than any other two membranes within the cell, right? And then between the cis cisterna of the Golgi to the one of the next cisterna, these two membranes are similar, and so on and so forth. So that's why these vesicles have to go through each cisterna and then go out toward the plasma membrane. That's why this vesicle cannot skip all the way to the plasma membrane, because the two membranes are too different for fusion. Now, can there be a case where you have a misguided vesicle and it moved to the wrong place? Yes. But what happens then? No fusion, because the membranes are not similar enough. That's really a very important concept to understand. So whenever we talk about fusion, and later on, let's see, have we phagocytosis or anything like that, where you have fusion into like lysosome, you have to think always that whenever two membranes fuse, that means that they have they are reasonably similar. Okay, how similar is reasonably similar? I don't know. Just reasonably. And what does that mean by reasonably similar? We're talking about the lipid signature. We're talking about the type of phospholipids, as well as the ratio among all the phospholipids. That's what we're talking about. So that answers your question, I think. Yes?
You just answered it.
Oh, okay. Okay? So that's fusion. Once fusion occurs, actually this is another part of your question, is if you notice that if this were the non-cytosolic phase, and this is cytosolic phase, you notice that these two, after fusion occurs, it'll invert, right? So that right there will fuse in with this, and this right there will fuse in with this. So it just opens up like that, right? So the two here, and it opens up like that.
So it'd be similar phospholipids, right?
Right. So therefore, the leaflets,
Yeah.
the non-cytosolic leaflet is still connected to the non-cytosolic
Cytosolic,
leaflet,
yeah.
and the cytosolic leaflet would still be attached, fused into the non-cytosolic leaflets.
Okay.
And I think that's the second part of your question.
Yeah.
Okay. All right.
Makes sense.
We're good. And then this process occurs over and over and over again sorry to be back and forth. So in each of the organelle, a different type of translational modification may occur, right? And when different types of translational modification occurs, then what happens is that some of these proteins may move forward, but some of these proteins may be told to, well, the modification is done. Why don't you go back to the ER, because this is an ER protein. Then that's why you have movement of the vesicles in both directions, right? Does that make sense? So proteins only go and end up in certain cisterna of the Golgi as long as the required translational modification occurs. If translational modification is complete, right, then the protein will be delivered to where they're supposed to. Now, if the protein has to go out to the plasma membrane or lysosome, then definitely they will have to be carried to each cisterna all the way out, right? So again, there's no skipping in line. Okay, we're good. So at this point, I'm going to let me let's see. Right, the roll. Yeah, okay. So that leads into this, and I was going to talk about lipid trafficking, but we can do it afterwards. Okay. So then, just like I said, so the role of the Golgi apparatus mainly, there are a number of different covalent modifications that can occur, but definitely glycosylation is the most important, right? Along the way, all the way until you get to the trans phase of this, the Golgi. So the trans phase of the Golgi is really important because this is where proteins, different groups of proteins are sorted. So let's say proteins that are to be secreted are gathered in one place. Proteins that are to be delivered to the lysosome are gathered in another place, right? So they're sorted and then put into the vesicle and then transported to the various areas. So there's a mechanism within the trans phase of the Golgi that will do all of that. Will sort and make sure that these proteins are delivered to the right places. Okay? So that's what I just said. Just an interesting fact. There are over 100 enzymes that carry out glycosylation. So and they're not all in the same Golgi, the cisterna. They're not all expressed in the same cell even. So then many of these are cell specific. Okay, so that each cell would express a certain subset of these glycosylating enzymes. What does that have to do with anything that we talk about? Pattern of gene expression and also recognition, right? Carbohydrates on the surface of a cell can be used for recognition. So that's how one cell recognizes another cell, because that cell will only express a certain pattern of glycosylating enzymes. Therefore, only a certain pattern of carbohydrates would be on the surface of a particular cell, and so that cell is recognized as so because of the pattern of gene expression, right? So we're going back to the pattern of gene expression. So, we have over 100 genes that encode for over 100 enzymes. But not all cell would express all 100 enzymes. Okay, so this is where I'm going to go back a little bit. So there was a question in the other section, and we should actually consider for this section as well, is the idea of membranes, right? So we looked at proteins. Now let's look at the membrane part. Let's look at the trafficking of the lipids. As the vesicles are being pinched off, right, carrying the proteins, that means that lipids are being removed. Does that mean that the membrane gets smaller and smaller and smaller? Relatively, if you have one or two vesicles, it won't, because the difference between 999,999 from a million is nothing, right? Right? But if you have enough vesicles that are being moved, then yes, you will have a reduced ER. And if enough moving that way, you will have a reduced cisterna, right? So that's one question, is that how do you then fill it back up? Second question is the idea of of lipid signature and protein signature. You're not only removing and you're reducing the size of the membrane, but you're also messing up the lipid and protein signature, right? So then, how does a cell deal with both? How does the cell deal with keeping the size of the organelles constant? Is by what I just said earlier, is by having the vesicular transport occurring in both directions. So if you lose a bunch of phospholipids going that way, then get replenished going this way. And again, because these two membranes are similar, right, then getting dumped in here or getting dumped in here, it's not going to vary the lipid signature that much, or the protein signature that much, or the cytosolic submembrane protein network that much. Does it vary all of those signatures? Yes, but not that much because they're similar. Does that kind of, right? That makes sense, okay? So that is in terms of the size. Now in terms of messing up with the lipid and all of the signatures, and how do you recover that? Does it vary that much doesn't mean that it doesn't vary, right? There's still variation. So how do you recover to get this precise lipid signature, precise protein signature? Yes, sir?
Are vesicles, are the signatures of vesicles modified so that they can recognize certain target compartments?
Signatures of certain vesicle? No. The vesicle that comes up, that vesicle right there, has the exact same lipid, protein, and submembrane meshwork signature as the ER, but not the same, exactly the same as a cis-cisterna. So all these signatures are different. All organelles have different signatures. So I'm not sure exactly, and we've said that, so I'm not sure what the question is.
I was wondering if on the way, as that vesicle is traveling, if the makeup of the lipid signature is modified at all.
I think I see. You should not think of this vesicle as the vesicle is traveling. The vesicle doesn't travel. The only place that this vesicle travel is to the cis-cisterna, and then a new vesicle is going to be formed. So when each vesicle is formed, the vesicle will resemble the signature of the original organelle where it budded off, right? So when you think of it that way, then no, there's no modification. So okay, it takes me a while to understand the question. Yes, sir.
Are these co-proteins being reused, or are they just being degraded?
They are being reused, and they have a pretty long half-life. That's a good question. And if I have time, these coat proteins are actually pretty cool to how they bud off, but we don't have time, so. In terms of replacement of lipids, that's fairly easy, right? If you have in both direction, vesicular transport going in both directions, that's easy. Okay. How about recovering of the protein signature first? That's fairly easy. If a protein, let's say a protein that must be here, that gets stuck in this vesicle that move out toward the cis-cisterna, guess what? It's going to be gathered and delivered back here through vesicular transport going the other way as well. Right. So that's easy. The problem is recovery of the lipid signature. Think about the fact that when you move the lipid in this way, you're moving the lipids in bulk. In bulk, that means a lot, and very non-specifically, basically. You just take whatever lipids that's here, you dump it into the next organelle, right? That messes up with the lipid signature. So therefore, in order to replace or recover the lipid signature in a very refined way, right? Very specific. If you only want 2 molecules of phosphatidylinositol, you better only have 2 molecules of phosphatidylinositol, right? If you have 3, then it won't no longer an ER, the lipid signature of the ER membrane. Does that make sense? So the particular phospholipid as well as a particular amount of that phospholipid is very important. Is very specific. So to be that specific, then to recover this lipid signature, lipids have to be moved one molecule at a time. So there's a system where one molecule at a time of a lipid that get moved from one membrane to the other. And in this case, it does not follow the same flow as vesicular transport. So a lipid from the ER, and this is where lipids are formed anyway, a lipid from the CER, let's say the lysosomal membrane needs a particular lipid, that lipid can be yanked out, removed, ripped out of the ER membrane, and placed into the lysosomal membrane directly. One phospholipid at a time. So that is also referred to as lipid trafficking. So lipid trafficking can be in bulk through vesicular transport or by movement of one phospholipid at a time, one sterol at a time. Does that make sense? So I came up with multiple questions, and I think I answered all those questions. And any question, Miss Ma'am, and then there and then there.
Does the same thing happen with the plasma membrane?
Yes. Yes, ma'am.
I'm sorry, I'm not understanding. Are you saying that if they move in bulk, they require vesicular transport, and if they're just moving one at a time, they're going directly where they need to go?
Yes. So through vesicular transport, lipids are transported in bulk. Back there, yes, sir, Wesley.
Oh, I was just gonna ask you to repeat the last couple of things about the lipid trafficking.
Okay, so let's say vesicular transport will transport lipids in bulk, in huge amount, and then to recover, to truly recover the lipid signature, then phospholipid especially, but other lipids have to be moved from one membrane to another membrane individually, so one molecule at a time. So both are considered to be lipid trafficking. Yes, sir.
Is that through some kind of protein?
Yes, through a protein called phospholipid exchange protein. And then we actually had about 3 different slides on this, but the concept is kind of difficult to understand. But think about the fact that if you remove the phospholipid from one layer, that means that layer is going to be smaller than the other layer, right? So then there's a process called flip-flopping. That's an actual scientific term. Flip-flopping is catalyzed by, guess what type of enzyme? A flipase. That can flip-flop phospholipids from one layer to the other. But anyway, so I won't go into that. But that's a good question. So it requires a very specific protein that can do that. Yes, ma'am.
Can you give a quick summary of what you said about restoring the precise protein signature?
Yes, to restore the lipid signature, one phospholipid at a time must be removed from a membrane and delivered to a target membrane.
The protein signature of the lipid.
Protein signature. To recover the protein signature. This has nothing to do with the protein signature. This has to do with the lipid signature. That the lipid itself needs to be moved from one membrane to another membrane.
Would you give an explanation before about how to recover the
Oh, protein signature. Okay, I keep hearing lipid in my head. I have voices in my head. Protein signature. Protein signature is recovered mainly through vesicular transport. So each protein, see, lipids don't have signal sequences. Proteins do. So if a particular protein needs to be moved, move to a certain organelle, all it has to do is show the signal sequence, and then it'll get gathered into the proper vesicle and delivered there. Good? Okay. All of this, and again, I'm just being redundant here. All of this requires lipid signature, protein signature, and sugar, and you know that all of these are asymmetric. That this goes along the same idea of if cytosolic, always cytosolic. If non-cytosolic, always cytosolic. And so therefore, the two the two faces or layers of a membrane are different, right? Different in terms of lipid signature, different in terms of protein signature, and different in terms of where the sugars are, and different in terms of where the cytosolic submembrane protein meshwork is, right? So they're all asymmetric. That means that one side is nothing like the other side. They could practically be two different membranes. Okay, they're not the same. They're the same membrane, but they have different signatures. So again, I'm just being redundant here. And we went through that. All right, we're almost done. So the idea of this, and again, this slide is really important. So memorize this slide, but then I'm going to go to the picture and show you. And the picture is kind of complicated, by the way, so I'm only going to focus in on what I need you to know, and not. So don't get mesmerized by the picture and then not see the particular things that I want you to see. Right? That's a huge picture. Okay, so here's what I want you to know is vesicular transport occurs, right? And then these proteins are going to be sorted, and then sent out to the various areas, right? So then a vesicle is going to get pinched off. That vesicle that gets pinched off, we were talking about the exocytotic pathway or the secretory pathway, either one. Secretory pathway is the same as the exocytotic pathway. Both of them are a part of the vesicular transport, right? That vesicle can be considered to be either a constitutive, can undergo what is called a constitutive secretory pathway or exocytotic pathway, or it can undergo what is called a regulated secretory pathway. What's the difference between the two? The difference between the two is that if you remember the term constitutive, that means always, right? Doesn't mean a large amount or a low amount. Doesn't mean spontaneous even. It just means always. You still have regulation. You still have to have, remember, coproteins inducing budding, transport, targeting according to protein and lipid signature, and then fusion according to lipid signature. All of those that's regulation, right? However, this is done continuously. This is done continuously without additional signals, right? So that's a constitutive secretory pathway. Whereas the regulated secretory pathway is when the vesicle is formed, it just stays there. It doesn't continue. Okay? It's only going to continue when this particular cell receives a signal that tells this vesicle to now move toward the plasma membrane and complete the secretory pathway. So, and I'll give you an example, a couple of examples if you want, but right off the bat, did everybody understand the difference between the two? One needs additional signal, and the other one does it constitutively. Yes, sir?
It's using the vesicle, the secretory vesicle just remains in the cytosol until
Right,
the
yes.
cytosol is divided to the
Yes.
extracellular.
Not only the cytosol, but within the cell cortex area.
Okay.
Yes, just remain in the cytosol. Yes, it's not as is. Okay. So example would be allergy, right? You have mast cells. You've heard of mast cells. Mast cells are referred to as granulated cells. Granulated cells because if you look at a microscopic picture of a mast cell at low resolution, you see that it's dotted. Where it's not really dotted, those dots are actually these secretory vesicles right here, right? So if you look at them, if you look at high resolution, you see that in the cell cortex, that you have a bunch of these secretory vesicles that are just sitting there, okay? So now you have an allergen. An allergen would come in, and we won't go into all of the tech immunology if you want to know exactly what happens, but the allergen then would send a signal to the cell to then allow these secretory vesicles now to move toward the plasma membrane and fusion occur too. So in other words, the allergen will pollen or maybe toxin of some kind will tell the cell to complete the secretory pathway. Right? It won't do that until it completes it. Okay. Another example would be your neurotransmitters. At the axon terminal, the same thing happens is that you have these vesicles, right, that are hanging around containing neurotransmitters. Now, when there's a signal, action potential, and so on down the axon, then these vesicles are going to be told to complete the secretory route. Okay. So. This occurs more often, and this is only in very specific cases like the two that I mentioned. Are there any questions about that? Yes, ma'am.
How long do the secondary vesicles stay there?
Pretty long. Pretty long. Think about, let's say, mast cells. That if you don't have an allergen, it could be as long as a mast cell is alive, which is months. 3, 4, half a year, and then when the mast cells die, then it dies. But when the mast cell is being made, then it starts accumulating these regulated secretory vesicles. In terms of the exons, these vesicles last even longer. Right? But they remain there, stay there for a while, and then the vesicles may actually go back in or get delivered to the lysosome. For instance, no, I won't go on it because it's a little more complex and it's too much time. But for a while, but it's not permanent. So the question might be: is it permanent? It's not permanent. Are we good with this? Co-translation. Another difficult, fairly difficult concept. If two proteins that are going to be made, right, and to be delivered to roughly the same place, and to perform a certain function together, then what happens is that they're not going to be translated and modified and delivered independently. They're going to be translated. The initiation of translation is independent, but then they somehow at some point come together and they will undergo modification, translational modification, as well as transport together. So we call that co-translation, right? So it's a misnomer. It's not really co-translation. It's co-modification and co-transport, right? Is, for instance, you might have collagen and integrin, right? Collagen and integrin, you know that this is a cell that is not an epithelial cell. There is a fibroblast, that this fibroblast is going to have to adhere to the extracellular matrix through collagen anyway. Then collagen is going to be synthesized in the cytosol, right? And integrin is going to synthesize and then pulled into the ER. But then the collagen and the integrin are going to be gathered in the same vesicles, right? Interacting with each other all the way from the area of the ER, going through all the cisternae, all the way out to the plasma membrane. Okay, and then we say that that collagen is secreted, but really it's not really secreted, right? Because it's already bound by the integrin. The integrin would just flip out, and then now you have the collagen that's going to be connected to other collagen. Does that make sense? Okay, so again, so the idea of really translational modification, packaging, transport, not really translation per se, but at least the modification and everything else they're joined together, and these are proteins that are going to end up in the same place anyway, and they're going to end up interacting with each other or one another and performing the same function anyway, right? So they're going to go through this whole process together. So when we talk about, earlier when I said something about proteins that are alike or gathered in the same place, that's what I meant, is this is a part of it. It's not just all the integrins will be gathered in one place, but all the integrin as well as associated proteins that are undergoing co-translational modification and co-transport are gathered in the same vesicle. Okay? And not only on the non-cytosolic side, but how about on the cytosolic side as well? Let's touch on a couple more organelles that belong to the endomembrane system, and then talk about the endomembrane system, the organelles that don't belong to the endomembrane system. The lysosome. The lysosome is where proteins and molecules go to get degraded, right? So remember a couple of lectures ago, I said that exocytosis and endocytosis are similar processes. So if we were talking about endocytosis, it's the same process except moving the other way. That's it. Okay. So the lysosome is where you have all these enzymes, right? All these enzymes. The lysosome is a very acidic. It has a very acidic condition. It's pH around pH 5. The lysosol is roughly physiological pH. And so here's the deal about pH is that the ER has roughly physiological pH, but as you look at the cisterna and all the medial cisternae and then the transcisterna, the pH start dropping as you move outward, right? And then vesicles that are being either secretory vesicles or endosome vesicles that are formed through endocytosis, the pH is about a pH 6 as well. So a slightly lower pH than physiological pH. So far so good? So how these organelles, let's look at just lysosome very specifically. How lysosome can have such a low pH condition is due to the proton pump. So far so good? So what happened is that the proton pump would take protons from the cytosol and pump it in, and that's how the pH is lowered. So far so good? But this is actually a more complex idea than it sounds. Where did this proton pump come from? It was translated in the cytosol, forward into the ER, and has to go through vesicular transport out to the lysosome. Then why is it that the ER lumen is not pH 5? Why is it that the Golgi, all the Golgi apparatus, the salinity of the Golgi apparatus isn't pH 5? Right? Well, to work around it, if it's not a lower pH, because the proton pump is not really working at maximum. Does that make sense? Right? So even though you have proton pumps everywhere, but they're not really, so let's say in the ER they're not working at all. In the cisterna they work a little bit, so that's why the pH drops a little bit. And then as you move out, more and more proton pumps are activated until you get to the lysosome. This is where practically all of these proton pumps are activated. Okay? So for the most part, so again, let's just stay with the lysosome. That's just the explanation throughout, but stay with the lysosome. How is the proton pump activated? Why is it that it's only in the lysosomal membrane before it's activated? It's due to lipid signature and protein signature of the lysosomal membrane. That's what activates the proton pump. So the lipid signature and protein signature, as you can see, has this other function as well, right? right? So it allows for the proton pump to get activated. So it's just not for recognition, it's for activation as well. Okay? And it's an actual pump, so therefore ATP is required. Versus something like an ATP synthetase where proton flows the other way and the ATP is made, right? And that's the electron transport chain and oxidative phosphorylation. So that's the lysosome. Yes, sir?
You said that the proton pump has to be transferred through this particular transport, right?
Yes, the pump itself has to. And how about these enzymes? They also have to go through. So why don't they work in and this is just a simple chemistry problem, is why don't they work in the other organelles as they're being transported? Because their optimal pH is pH 5. So they actually might do some damage along the way, but not much because the optimal pH is pH 5. So that explains both the proton pump and these other enzymes. Don't try to memorize all these enzymes, but it's just nuclease means that these are enzymes that digest nucleic acid, protease that digest protein, and so on and so forth. Okay. And that's what I just explained. Right. So specifically, it's that protein right there. But if you just understand that it's the lipid and protein signature, that's all that. Very general is all that you need to know. Yes, sir.
When lysosome digests another particle, how does it know which enzymes to use, or does it just, like, does it know by when it binds to whatever it's gonna, does that would tell it, okay, release this enzyme is required?
Right. So it's all binding. Right. So a nuclease would never digest a protein, for instance, if I understand your question correctly, because a nuclease will look for phosphodiester bond, right, versus looking for a peptide bond, for instance. So it's all binding. By education and by research, I'm truly a protein chemist, and I think of everything down to the level of binding, down to the level of electrons and electrons. That's in my head, that everything boils down to electrons, boils down to size, shape, and charge. Okay, so let's talk about the endosymbiotic theory. We won't talk about the endosymbiotic theory, but we'll talk about organelles that came from that, mitochondria, and the mitochondrion and the chloroplasts. Okay, how do they work? More specifically, the mitochondrion. Okay. So remember, the mitochondrion used to be millions of years ago, used to be a prokaryotic cell, therefore it has its own DNA, therefore it can carry out its own metabolism. Okay? It can even divide on its own by binary fission. Okay? But the two membranes are different is because the inner membrane is very similar to a prokaryotic cell, whereas the outer membrane is not. The outer membrane actually really originated from the eukaryotic cell, right? The host cell, the host plasma membrane actually. Okay, so just the different component, the matrix versus the intermembrane space versus that. So then, this is an important thing. So lipids, all lipids of the mitochondrion, at least in the inner mitochondrial membrane. Now, so for mitochondrion, you have to separate between the outer and the inner mitochondrial membrane. For the inner mitochondrial membrane, all lipids are provided by the mitochondrion itself. And just about everything I say about mitochondrion applies to chloroplasts, by the way. So all lipids are synthesized by the mitochondrion itself. Most proteins, isn't this interesting? Most, but not all proteins are encoded by the mitochondrial DNA, but not all. So there are mitochondrial proteins that are encoded by the host, the nuclear genome. So the whole idea is that, for instance, you know that glycolysis occurs in the cytosol, right? And the Krebs cycle and electron transport chain and oxidative phosphorylation occurs in the mitochondrion, but a prokaryotic cell actually can also carry out glycosylation, but not a mitochondrion. The point is that after the mitochondrion had survived in the host cell for a while, it got lazy. And so some of the activities that the host cell is already doing anyway, that through evolution, the ancient prokaryotic cell would have lost those activities. They lost those activities because they somehow lost their genes due to mutations, due to a number of different things, right? Due to horizontal transfer, probably from at the time during evolution from the prokaryotic cell to the host cell. So let's say glycolysis, for instance, right? So the ancient host cell actually used to be anaerobic, so they don't carry out. They don't make a lot of ATP. Anyway, so I won't go back that far, but basically the idea is that the mitochondrial DNA does not encode for all of its protein. Okay? That some proteins have to be delivered in, and that would bridge on to the next segment. But all lipids do for the inner membrane. Where do the lipids for the outer membrane come from? The host cell, through vesicular transport. However, why is it that the mitochondrion then not considered to be part of the endomembrane system? It kind of is, but we don't call it that way, but it kind of is because lipids still need to be delivered there. Does that make sense? So you might learn by definition that, okay, so you don't have, sorry, it's taking longer than I thought. You might learn that, the mitochondrion is not a part of the endomembrane system because being in the endomembrane system, that means that the membrane can flow from one organelle to the other. But guess what? Membranes also flow into the mitochondrial membrane. It's just it's the outer membrane that it flows into. Has nothing to do with in the membrane. Okay?
So it's just spontaneous as well?
It's directed. It's not spontaneous.
Okay.
It's constitutive, but it's not spontaneous.
Okay.
So that's what that says right here. Okay? And there are proteins that have to be delivered there as well. So proteins that are encoded by a nuclear gene that are expressed and translated in the cytosol, how do they get into the mitochondrion, right? They get into the mitochondrion through the twins, Tom and Tim. Translocase in the outer mitochondrial membrane and translocase in the mitochondrial membrane. So I call them the twin. So Tom is on the outer mitochondrial membrane O, and Tim is in the inside. And what Tom does is Tom pulls very similar to the ER membrane, pulls the polypeptide in, okay, and then Tom can actually insert the protein into the outer mitochondrial membrane if it wants to, if there's a signal, a topogenic sequence, or it can hand off the polypeptide to Tim, and Tim can actually also has two options, either insert the protein into the inner mitochondrial membrane or actually allow the protein to become soluble. I don't need you to know a lot of details about this. I just need you to know the fact that there are proteins that can pull, there are membrane proteins in the mitochondrial membranes that can pull polypeptides that were encoded by a nuclear genome. Okay, number one. Number two is that only these proteins can be pulled in and be placed in the outer mitochondrial membrane, in the mitochondrial membrane, or in the matrix. So they can be anywhere. However, the reverse process doesn't occur, so that mitochondrial DNA can only express either soluble proteins in the matrix of the mitochondrion or in the inner mitochondrial membrane, but the proteins are embedded in the outer mitochondrial membrane were encoded by the nuclear genome. So the reverse is not true, right? These proteins that are encoded by the nuclear genome can exist anywhere in the mitochondrion. But for mitochondrial membrane, the genome, they only encode for proteins that are either soluble in the matrix or or in the inner mitochondrial membrane, but not the outer mitochondrial membrane. There's no way of delivering out. Okay? And again, anything that I say about the mitochondrion applies for chloroplast. Related activities, related functions for vesicular transport, or endosomal system meaning vesicular transport. Related activities include, like as I mentioned before, endocytosis, either phagocytosis or pinocytosis. Also for removal and recycling of membrane proteins. So if you have a membrane protein that were delivered to the plasma membrane and just sit there for 3 months or so, or the cell actually if it's underused as well. So proteins can be recycled if it's overly used and then it needs to be replaced, or if it's underused as well. If a cell decided that protein was made but it's not doing anything, why is it sitting there? Then it'll get pulled in, right? So it'll get recycled through endocytosis. And then EM turnover and remodeling. So the extracellular matrix, so the cell would sense its extracellular matrix and decide either that the extracellular matrix needs to be recycled or that conditions have changed and the extracellular matrix needs to change. Then the extracellular matrix will also be endocytosed, right, digested, and then new extracellular matrix material will be delivered. It's very complex, right? So it's not just moving stuff in within the cell. The cell can sense its environment and can change its environment if it chooses to. Okay? Yeah, we already discussed this, right? This is. I'm not going to require you to know any of this. Okay. So last. So what I'm going to do is for this last one, it's been 3 years in the making, and we still don't have a picture. All we have is this. Okay. So I'm going to draw you a picture by hand on the board. Okay. So this is the only time that I'm gonna lecture on the board, unless actually in the last semester there were a few times where I had to do that because the system was down. So I'm gonna draw this, all of this. I'm gonna do all of this on the board. Okay. So we're not done, but I'm gonna do that. Let's see. And then sorry, that means I only have about 10 minutes to answer questions.
Yeah, I got it.
All right. So all of this to build, to allow a neuron. Here's a neuron. Right. Here's a neuron to interact with its target cell. So it is a presynaptic cell. Here's a postsynaptic cell. Let's say that you start out with the neuron not having an axon yet and not knowing what its target cell is. And so basically during neurogenesis, right, during the process of embryogenesis of development of your brain, or even as you're learning, hopefully you hear the term making new connections, right? So this is what it is how does a neuron make new connections as you're learning? Hopefully you're making new connections, right? The idea is that, remember, microtubule polymerization, the regulation of microtubule polymerization depending on where it is. So you want to direct microtubule polymerization here, not here, because if you do it here, the exon will move the other way. So you want microtubule polymerization and the regulation, so everything from nucleation to everything that we talked about, right? So that an exon can be formed. Okay, so And then exon, here's the exon terminal. And then what happened is that you have all these microtubules will also be used to deliver mitochondria, because you need a lot of ATP down here. Okay? And how about neurotransmitters? I just talked about neurotransmitters through regulated vesicular transport, right, or secretory pathway. But where did these, let's say, if they were proteins, or where did these neurotransmitters come from? All the way up here.
Okay.
Right? So proteins, so neurotransmitter, Golgi, neurotransmitters have to be put into vesicles, and then these vesicles then will travel down here, waiting to be secreted. There you go, right?
Okay.
How about making connections? How does it know that this is a target cell? Remember, again, I'm bringing back everything that we talked about because this is supposed to bring back everything that we talked about for this set of exams, right? Is remember scaffolding. Remember that these two cells need to hang on to each other, right? So then there are proteins here and proteins here that hang on to each other, and there are a bunch of proteins, including receptor for neurotransmitters, which is scaffolded on that side.
Okay, that makes sense.
Does that make sense? So again, I'm trying to put everything that we learned together for this set of exam. Okay, and then these, and then they'll talk now. So when you learn, so move forward to when you took, you learned about action potential in what, 5th grade or in systemic phys. You only start from here, right? You don't start from what I just showed you. Okay, so what I showed you is how this is built. Okay, so we're really calling this hardwiring. Again, you hear me say that term all the time. Hardwiring meaning that everything, all the components have to be in place. Okay, so that when that cell receives a signal, it would send action potential down here, and if it's a my brain telling my toe to wiggle, then you would have myelin sheath all over the place, and myelin sheath would not have a lot of protein, so therefore you will not have action potentials here. So instead of having, let's say, 1 million action potential, you only the exon only carries out 1,000, let's say. And then 1 million, if it takes, let's say, 1 millisecond per action potential, I'm just making up number, then 1 million means that it actually takes 1,000 seconds, right, for the message to get here. 1,000 seconds, that's a long time. If I have to wait that long for my toe to wiggle, right, that's people with MS, that's what happens. Okay, because they don't have, and the myelin sheath, what it does is that it allows for action potential and then skip through, right, and then have action potential again. So skipping through these allows for the signal to be passed on faster. How are they skipped through these? Because of the lack of proteins, lack of ion pumps. So again, everything that we talk about, right? I'm referring to the protein lipid ratio. If you have a very low protein lipid ratio, then chances are there's not a lot of activities, right? In this case, only in this particular case, what we're talking about is the absence of ion pumps. So if you don't have ion pumps, then you don't have an action potential, then the signal will move through. And those of you who have taken systemic phys, you know that the sodium ions actually diffuses across there, right? And all of that, so I won't mention that. Okay? So now you have this. That signal, more stuff. Remember sequestration of calcium, right? Calcium is sequestered in different places, including the extracellular matrix and the ER, but the ER is all the way up here. There's no ER here. So the calcium that is used is actually the extracellular matrix at this end. And what happened is that signal transduction pathway, guess what it tries to tell the cell to do? To activate this calcium ion channel right here. Okay? Calcium flows in. Calcium then will bind to these vesicles to tell them to complete the process.
Okay.
for allergy for mast cells, it's similar except the amount of cyclic AMP is involved, and it's a little more complex, but not much. In this case, calcium literally binds to membrane proteins on these vesicles, and then to direct them to go toward the plasma membrane, releasing neurotransmitter, and then that's how that works. So everything that we talked about is in this example. Okay, does that make sense? Hopefully that actually put things in perspective for you. Yes, sir.
Forming that axon, is that gonna be something like that, was it the centrosome?
This is we are not completely sure, because here's the reason why. Because we know that for exons, actually, for exons, the centrosome is used, right? So that's the centrosome right there. That it does radiate out. Number one, there's a lot of, the exon is pretty big, so there's a lot of high concentration of microtubules moving that way. The other problem that we know, at least I know of, that hasn't been resolved is the fact that microtubules actually is built in both directions. So it's not only being built in going to plus and toward that way, but also in this direction as well. And how is it built in this direction? I don't think people know exactly. So partly the answer is yes. Partly it's from the centrosome. Partly it could be from basal bodies that are found along here. But that means that are there basal bodies from here that go back this way? I don't think people have found any basal bodies that are in the exon terminal that I know of. So this is, believe it or not, this is an area that we still don't know as much as we should. So that's a very good question. Okay. Yes, sir.
You mentioned MS. So is that because as they lose myelin, the pumps basically stop working?
No.
You're saying, why do the action potentials? Why are they not?
These myelin sheath are formed from Schwann cells. Schwann cells that then the plasma membrane of Schwann cell would form. For MS patient, the Schwann cells are not made, or there's a problem with the Schwann cells. So therefore, the myelin sheath aren't there. When the myelin sheath aren't there, instead of being able for the sodium to diffuse across a large area of this exon, they have to go through action potential in that area. And that's what happens. And what does that have to do with your muscle? Well, if you don't exercise your muscle, you use it or lose it, basically, as far as muscle is concerned. Okay, and so that's why people with MS, then the wasting of the muscle it takes a while, right? Whereas MD, which is a muscular dystrophy, which is a problem with the actual muscle, the formation of muscles themselves, it happens, you see, in earlier children, so earlier, right, faster or earlier. So it has to do with that. So both of them have to do with your skeletal muscles being unable to contract, but one is due to the actual contraction process, and the other one is due to the neuronal process. And so that the timing. So for instance, when I teach immunology, that is, that timing is everything. So people with different diseases that have the same symptoms, but sometimes it show up early, sometimes it show up late, is because of that. Is because they have the same symptom, but due to different causes, and depending on the cause, then that leads to whether or not you have that disease early on or later. Yes, ma'am.
Can you explain the role of microfilaments in this example, or?
Microfilaments? Not much. Microfilaments should be in the cortex here, and maybe if there's refinement, if you have a vesicle that needs to, this vesicle needs to secrete neurotransmitter here, it might actually be moved over a little bit. So just for the microfilament in this case would just be for delivery, for transport. Right. Yes, ma'am.
Can you repeat the purpose of the calcium ion pump or the channel?
The action potential would open up the calcium ion channel, which allow calcium from the extracellular matrix to flow into the cytosol. With an increased amount of calcium, then calcium would bind to the membrane of the regulated vesicle, secretory vesicle. Then that will induce these vesicles to move toward the plasma membrane. So basically, basically this is what is this saying is that the calcium then would I know that people know exact. I'm just guessing here. I'm guessing is that it would then activate the molecular motor to then move these proteins over. Either molecular motor or activate the Rab protein. I know that people know, I just don't know, right?
Okay.
So, but the calcium actually binds to membrane proteins of the vesicle here and drive the process forward.
Okay.
But it's stalled here because at this point, for these you would think, well, why would these vesicles not move forward and then the vesicles from the constitutive secretory pathway move forward? Because of the lack of recognition of some kind of a protein signature, or maybe the cytosolic submembrane protein meshwork, and then the calcium then would open up that signature.
Okay.
5 minutes of study session if you want. Or go home. Question? Yes, sir.
You said that the calcium is analogous to